expensive. That's the branded drugs. But as I have said, especially in Kenya, we do have a problem. We do have a weakness here and there. And when a drug is patented, at no time does that original manufacturer, the original manufacturer is the one who goes out of their way and does a lot of research. They do the clinical trials, they do a lot of research, they even research on the dissolution, everything. They research on the good binders. So even when now they come and release a molecule into the market, they release the ciprofloxacin molecule. There are some tricks they don't tell you. They will not tell you which binders they use. They will not tell you the surfactants they use. So that is where now these different drugs have differences in their bioavailability, in their dissolution, because the one who did the initial tests made sure that they went out of their way and did all these tests, tested with different binders, with different surfactants, and all those other mixtures, colorings, and everything. So their ultimate drug is a well-tested drug when you talk about the norfloxacin and then the ciprofloxacin. Now, what they sell is a molecule. So they will give you the ciprofloxacin molecule, but how to make it into a tablet, that is their trick. That is what they eat from. So now when these other people come to make the same molecule, they just go and copy. They go for the leaflet for ciprofloxacin. They don't do any test. Some of them don't do any testing. I believe in some of the countries they do testing, but in some of the countries also, they go for the leaflet. Like in Kenya, I believe they buy the norfloxacin powder. They come mix it with binders, mix it with surfactants. They do not do any testing, as I have said, and I will say that without fear, that some of our institutions are very corrupt. So when they take these drugs for testing, they're supposed to be there's a lab. There's a lab, national something, call it something, control lab. Then of course they will follow with with a lot of money. So these drugs are never properly tested. As I have said, remember, they do not know the binders, they do not know the surfactants, they do not know the coloring. Those are hidden by the original people who manufactured those drugs. So these guys who are synthesizing the generics or manufacturing the generics, they don't do proper testing, because if they did the proper testing, they will also find out which of the binders will work well and so on. And that is where I said some of these drugs will have a lot of differences. The original product and the generic will have a lot of problems or a lot of differences because nobody researched, to be honest with you, like in our country. Some of these drugs are made in the back streets and nobody bothers. Nobody, just like in everything else we do in Kenya, nobody is bothered. Our 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 pharmacy and poisons board sometimes also is compromised, and therefore you can have a lot of, also even from other countries, you can have a lot of counterfeits. Counterfeits are those products whereby they are claiming to have a certain active ingredient and indeed they don't have. So you do have, because of the dangers that were faced with the quinolones, you do have some tests that you can do so that you can check on whether a product is a counterfeit. That means it looks the same but does not have the active ingredient in it. And that is the only I want us to look at those those tests that can identify whether a drug or the fluoroquinolone is a counterfeit or is indeed the actual drug. And counterfeiting, of course, has brought, especially when you do counterfeit of antibacterial agents, you can imagine we are giving a counterfeit to a patient with TB and they're already on second line. So they already have more drug resistant TB. What are you doing to them? You're killing them. And as I have said, it is the danger. It is us as the professionals who have refused to do our job well. And we are totally compromised. And being compromised, of course, we are allowing importation of counterfeits. We are allowing even those counterfeits to be produced within our country because we have eaten, and we do not care the danger that, of course, that brings to the patient. So I want us to look at the way, the identification or the screening, how we can screen. We'll have to look at them more. How we can screen for counterfeit fluoroquinolones. And one of the tests we can do, oh, I hope it doesn't shut down. That's what it's doing, and then it shuts down. So one of the things we can do is we have oxidation of ferrous ammonium sulfate solution. And so we make solution A as a ferrous ammonium sulfate solution, 0.1% weight by volume in 1% high sulfuric acid. So our solution A is ferrous ammonium sulfate solution, 0.1% weight by volume in 1% sulfuric acid. And what is the procedure? To the solid sample, 0.5 ml solution is, that is the ammonium sulfate in sulfuric acid, was added, followed by sodium bicarbonate to effervescence ceased. Development of a yellow-red color indicated presence of a fluoroquinoline. The blank had no color. So if you have a fluoroquinolone, in the lab, you can't determine whether it has any active ingredient or it is indeed a fluoroquinolone or counterfeit by addition of ammonium sulfate in sulfuric acid, and then you add sodium bicarbonate, and we are calling it the rapid color test identification, and there will be a color development, and if it is a counterfeit, there will not be any color development at all. Again, this is important, as we have said. The fluoroquinolones, especially initially, because the original brands were extremely there is Noroxin. Noroxin was the original one. And of course, we have ciproxin, as I've said, they were extremely expensive. So many people came with counterfeits. And of course, they want to reap from where they have not sold. But that's what happens. Then we have this, the second kind of test. You have that's the first test of identification. That test of identification for the fluoroquinolones, again, identification of the fluoroquinolones is whereby you make your solution A, and this is a solution A test for fluoride, where we test for the fluoride, and the solution A consists of, I think this is zirconium EDTA, zirconium EDTA PV, and zirconium EDTA PV, zirconium ethylene diamine tetraacetic acid pyrocatical violet. So it has a violet color. And that's what we are calling our reagent A. Zirconium ethylene diamine tetraacetic acid. The EDTA is a chelating agent. So zirconium ethylene diamine tetraacetic acid pyrocatical violet. And that's what we are calling our solution A. The acetate, then you make an acetate buffer of pH 4.2, was also prepared by dissolving 3.7 ml glacial acetic acid and 2.177 grams sodium acetate trihydrate in 50 ml. Again, remember, when you're making buffers are made by using the acid and its salt. So it makes the acid and its salt with a strong base. So we are combining acetic acid plus sodium acetate, and that makes our acetate buffer. Then you have your solution B. So that is a buffer, because we have to buffer the solution, because the test, of course, is most effective at a particular pH. Then solution B, you have 0.02 grams zirconium. This is zirconyl chloride, 0.02 grams zirconyl chloride, 0.03 ethylene diamine tetraacetic acid, and 0.0001 pyrocatical violet were dissolved in water. To this 25 ml solution A was added. So you take this compound. So you have this solution ethylene diamine tetraacetic. So this is almost like this other solution there. So it's like making how this solution is made. So you have 0.02 zirconyl chloride, EDTA, pyrocatical violet were dissolved in water, and then to this solution, 25 ml solution A was added. Which is our solution A? I think that is a bit confusing. So our solution A is test fluoride, zirconyl ethylene diamine tetrachloride. That is a solution A. I think, no, sorry. This is solution B. So this is solution A. The buffer, I believe, is our solution B. Then I think our buffer, I want to see that's correct. The buffer is our solution B, and the zirconyl, where the zirconyl is, I want to say, is our solution A. Let's see whether it makes. So then you take 0.02 the corneal, all that, and volume was made up to 100 ml. This reagent is stable for. So where have we added what? Let me see. So we have solution is a test solution. Then you have that is our buffer, and then you have 0.02 zirconium chloride plus 0.03 EDTA and 0.01 pyrocatical violet were dissolved in water, and that is what is making our solution A. To this 25 ml solution, this should be solution B was added. Otherwise, this is the same as solution A, isn't it? Solution A is the zirconium, which is that is a zirconium. Then there is EDTA, and then there's a pyrocatical violet were dissolved in water. To this 25 ml solution A was added, and solution A has 4.2 was prepared by dissolving glacioacetic acid. Where is even the drug? Then to this the added volume was made to up to this reagent is stable. Let us look at the next step. So we have our solution A, which has the solution. We have our solution A, which has a zirconium, whatever, and we have the solution, I think, B, which is, I want to believe, is our buffer. Then let us look at the procedure. The drug sample was fused with 50 ml of sodium nitrate in a wide mouth glass tube. After cooling, 25 milligrams of ammonium chloride was added, and heating was resumed to effervescence. So you have your drug sample, and then you put 50 milligrams of sodium nitrate, and then you heat. When you talk about you heat them together, the drug plus sodium nitrate, you heat them together. After they cool, you add ammonium chloride was added, and again you continue heating until the effervescence ceased. After cooling, the solid mass was dissolved in 0.5 ml water, and 0.5 ml solution B was added. The color change of solution B, so the color change of solution B from blue to yellow indicated positive test for fluoroquinolones. So I think what we are calling our solution B is this test solution. So here there is, I think here there was a little miss. This was not solution A. This is our solution B, is the zirconium solution. But all we are saying is that you must also add your buffer. I don't see the procedure that they are saying we add the buffer, but I believe when you dissolve the drug in sodium, confirm it, you dissolve it in sodium nitrate, you heat it, and then you allow it to cool, you add ammonium chloride, and then heat again. After cooling, the solid mass was dissolved in 0.5 ml water and 0.5 ml of solution B. And our solution B is the zirconium chloride mixture with ethylene, diamine, whatever. But we should have also noted that in this solution, of course, this solution must be buffered. So we must buffer it with our solution A, and our solution A is where we are calling the acetate buffer. That I think I've missed out. So we are saying, again, I think it's now clear. We take our drug sample and we fuse it, fusing meaning heating, with sodium nitrate in a wide mouth glass tube. After cooling, we add ammonium chloride was added and heated heating was resumed to effervescence. After cooling, the solid mass was dissolved in 0.5 ml of water. So you heat all that and you get a solid. You put it in 0.5 ml water and 0.5 ml solution B. And of course, solution B is the one we are calling the zirconium, is the one which has the zirconium chloride. That is the one, zirconium chloride. And then, of course, you will buffer. You must buffer the solution with the acetate buffer so that you can get the proper reaction. And obviously, the color change of solution B from blue to yellow indicated positive test for the fluoroquinolone. If there is no color change, then again there will be, it's a counterfeit. It will not have it means it will not have the drug. But you can't see this zirconium. We are calling it zirconium ethylene diamine tetraacetic acid pyrocatical violet. I was expecting it has a violet color, but perhaps now once you fuse, see, you have fused the drug and then you have added the solution B. I think that's where we are saying change of solution B from blue. I would have expected from violet, from blue, but let me confirm. I would have expected, because why are they calling it violet? I would have expected, but I will confirm again that the color change of solution B from violet to yellow indicates positive test of the fluoroquinolone. But I hope you are getting the idea. We are the zirconium EDTA and zirconium ethylene diamine tetracetic acid with the pyrocatical violet. So could be, I will confirm, as I've said, I have not looked at it. And perhaps the pyrocatical is the one which is violet. Perhaps when you make the whole of this solution, it turns blue. I will confirm. But the way they are calling pyrocatical violet, it could be the pyrocatical, which is called pyrocatical violet. So when we do the zirconium, ethylene diamine tetracetic acid, and we add the pyrocatical violet, the resultant solution is blue. I believe that is actually the proper procedure. And that solution with our acetate buffer, and then now that is the solution we are calling our solution. Of course, there is this way we are making it, and of course we buffer it. That is the way, that's the one we are calling our solution B. Then now that solution B, we have to prepare again our drug in a very specific way. Add sodium nitrate, fuse it, add ammonium chloride, continue heating until effervescence, and now that solution of the drug, that when once you heat it until effervescence is finished, it also means it's forming a solid. So you take that, you cool it, you take that solid, and you mix it with the zirconium, or the solution which is having the zirconium, ethylene acetic acid, and the pyrocatical violet. You put all of them together, and you will have a change of color from blue to yellow. That will tell you, or that will indicate the presence of a fluoroquinolone. If the color does not change from blue to yellow, again, it's a counterfeit. I hope that is clear. And I believe that is the way it is to go about it. Your pyrocatical is a violet. No, the pyrocatical is also a solution on its own. The ethylene diamine tetracetic acid, ethylene diamine tetracetic acid is a solution of its own. And then, of course, you have your zirconium, of course, as a metal. So you add your zirconium, you add your ethylene diamine tetracetic acid, you add your pyrocatical violet. And once you add all that, and of course you make it, you add it like this. They have said you take 0.02 grams zirconium. This is now called zirconyl chloride. You add your EDTA, you add your pyrocatical violet. You put all those compounds together and were dissolved in water. And then, yes, then you buffer that solution. You buffer that solution, and I believe the buffer is the solution A. So there was a little bit of a mixture here. So the solution A is our buffer, our solution B is the one which is zirconium. So 25 ml of solution A was added. So you're saying you take your zirconyl in this amount, you take your reagents in this quantities, and they'll give you the solution B, and then you add the buffer, because you must buffer that solution for it to give us good activity. And we are saying that solution can be stable for 2 weeks at room temperature. So now, once you heat your drug, or you prepare your drug the way we have said, then you take your solution B, and you can see your solution B now. It's very clear here. It has been buffered. The solution B has been buffered. You add. Once you add, there will be a change from blue to yellow. If there is no change, it means your drug is a counterfeit. But how often do we have the opportunity to do those tests? Very, very rarely. So what we would ask for is that if we are professional, if we behaved professionally, all of us, at all the positions we are, if you are the company pharmacist, you are professional. But you see, the danger sometimes, I'll tell you this with honesty, you become strict and you go to the industry, I can assure you, you will not get the job. You will not work there. They will sack you. Kwenda nyumbani. So that is the problem, because of the fact that we are desperate, even as professionals. You need the job, and because not all of us are professional. If all of us decided that we will be professional. So even if you sack me and bring the other pharmacists will also be professional, then we can be able to stand. But we know some of us are easy compromised. So if you stand and you are strict, more often than not, they tell you don't report tomorrow. Then, if there was also strictness and professionalism in our regulatory bodies, where one of our regulatory bodies is the Pharmacy and Poisons Board, such that before they register, because remember, the Pharmacy and Poisons some of those, all of those things, is the one which registers all the proper drug products in the market. So if they were completely strict and they are supposed to ask for all the clinical trials, all the in vitro trials that have been done for that drug, if they were strict and they asked for all those tests, of which I can tell you they are not. If they were strict and asked for all those tests, showing that the clinical trials have been done, the in vitro trials have been done, the toxicity tests have been done, then, and they look at those tests before registration of that product into the Kenyan market, then we will be able to control resistance a lot. But that sometimes, I'm sorry, is not happening from the manufacturing company. There is compromise. If you if you stand your ground and you are strict, as I've told you, and I will be honest, utaambiwa uende nyumbani. So unasema sasa, mimi niko na familia, I need the job. Then when it comes to the regulatory bodies, when it comes to the professional pharmacists who are dispensing the drugs, sometimes you're looking at a product, you can see it's already molted, you can see it's already, it has already, its friability is very low, it has already broken into pieces, but you cannot, unfortunately, most of us cannot stand our ground and be able to be heard and speak and with honesty and say, this is 40. Most of you, even when you go for your attachment, again, I want you to understand that you will go to some of those companies and you will see how compromised they are. When they get a product, you know what those SOPs you are going to do them in pharmaceutics. Of course, they have the in-house testing, but there is the final testing of that product to ensure that that product meets the pharmacopoeia standards. I'm telling you, we were also there. You test that product, it fails. Pitisha tu. Pitisha. So there are so many loopholes, there are so many gray areas, and that is why quite a number of these drugs, even when they are released to the market, you will find patients are taking them, but there is no activity. And that is why for those who have the money, they will go for the branded product because that product has been well studied. And of course, that product has a very high price because those people who make are trying to recover or recoup the money they used in the research process. That is why when you go out in the market, amoxicillin original will have a much higher price than amoxicillin, the generic. As we continue training you as pharmacists, before there were very few of us in the market. As we continue training you as pharmacists, we are asking you when you go to them out there in the market to do whatever you can do to improve the products that we are getting out and giving to our patients. The numbers matter. When we were being trained ourselves, we used to be 30 in a class. After every 4 years, you only used to be 30. Numbers matter. From that time, which is many years, it's the 80s. I think training of pharmacists started in those years of 80s. We do have many, many pharmacists. And I think if all of you could speak with one voice, if of us could speak with one voice, I think we can be heard. Because it is a high time, be it our regulatory body, be it the manufacturers of drugs, be it those who are storing them, be it those who are importing the raw materials, follow the law to the letter. Because we are exposing our patients, we are exposing many of our patients to substandard drugs because of money. Of course, when you go out there in the market, you will also realize that we have facts. Everybody is a doctor, everybody is a pharmacist. And therefore, some of the mistakes that are happening are not happening because of the professional pharmacists, because so many people want to work or to pretend they are pharmacy. But again, that is a failure of the regulatory body, because the regulatory body should be able to go out there and disregister all close chemists or close facilities that belong to people who are not licensed. But again, exchange of money, exchange of money. Some time back, I don't know whether that has changed, and of course, that I believe you are going to do law and ethics. Some time back, the people they used to choose, or the people who used to be appointed to be drug inspectors, used to be people with no training in pharmacy. I don't know whether that I need to check. I don't know whether that has changed. So these people, again, no training in pharmacy. They are the ones who are going to inspect the premises, close down the pharmacies. What do they know? They know nothing. So when they come, they are told, don't close. I can give you 50,000. I can give you 100,000. Fair, give me the money. So as I will repeat again, because there is increase in numbers, there is increase in numbers in terms of the you as a professional pharmacist, and especially young tax. It is the high time we streamline our profession, and numbers count, because numbers can be more forceful. Big numbers can be more forceful than small numbers, because some of the drug resistance that we are observing out there, it is a creation of us, whereby what we are what we are saying this, what we are manufacturing is already substandard. Some of the drugs we are allowing to come into the country are actually counterfeit. And I'm aware you know that. A lot of counterfeits coming into the market, a lot of money exchanging hands, and of course, a lot of resistance, a lot of drugs, and so on and so forth. So, my question is, when you graduate, or even now, what will be your role? What role will you play when you get out there? Because you can get out there, work in the industry, work in the regulatory body, work as an academician like us. There are so many areas. Work in the hospitals, work in the retail market. What will be your role? Will you allow yourself to have voice that can be heard, or will you be silenced like many of us have been silenced before? Or will you, all of you, will you come out and be able to say, let us protect our profession, let us save our profession, let us be jealously guard our profession, and therefore we will, because if we jealously guard our profession, we are assured of a job for many years. If we do not guard our profession, what we'll do is that there will be infiltration of many people who know nothing, and finally they will control the market. And therefore, our role as pharmacists will be of no consequence. So it is up to us. And that is why the training you are receiving actually is very important. It has really changed. There is a lot of clinical orientation so that you can be able, when you go out, to define the role of a pharmacist. That a pharmacist is a pharmacist who does the right thing at the regulatory level, at the manufacturing level, at the clinical level, at the academic level, and at all levels, that we have control of the drug. Because the unfortunate bit, the drug is a very expensive item. And because it's a very expensive item, it has brought a lot of room for corruption. And therefore, we as pharmacists ourselves have also allowed for the corruption, because we want to get rich quickly. But I do believe there is still room for change. There's still room for change, and the little change you make, you might think that little change you make does not have some influence. But hata ukichibua pahali polepole, unachibua polepole, finally you will get a way through. So whatever little you can do at whatever level, kindly and please do it. You might feel like it's not having any effect, but ultimately, little by little, joined together, ultimately will have quite a big influence on the practice of pharmacy. Otherwise, if we allow things to continue the way they are, we might be edged out, and nobody knows who will be practicing this profession. So with those remarks, it's my request that even when you do your attachment, because you must learn the language to talk to those people, because you cannot go telling them, hey, this product has failed. You people produce products that are substandard. You must have the etiquette, you must have the proper communication skills. But it's a fight. It is indeed a fight to make sure that the products, the drugs that are being released in the market, uh, have the correct or meet the correct pharmacopoeia standards. I think with that, can we sign in? Sign in, and I believe those that you have been told the same. I want to believe in the other units that as pharmacists, we must continue fighting, not being too aggressive, but fighting in the proper way by being well informed, by knowing our place in the healthcare system, so that we do make an impact. And that is why you find the pharmacy profession is now also leaning a lot on the clinical areas, so that you understand the influence of drugs on the patients, so that we have an edge, something that no other person understands out there. Of course, also we have, of course, that edge of understanding what these drugs contain and so on and so forth. But the danger is a compromise. Are you compromisable, or can you stand and defend your profession? That is up to you. That will be up to you. It cannot be up to anybody else. So can we sign in, please? Sign in, sign in, sign in. So I thank the Lord. I took some medication. In fact, I was waiting for azithromycin, but the cetirizine also has done a miracle. I actually didn't sleep because of the cough. It has actually controlled. I think it's an allergic reaction, so it has controlled the flu a lot. Thank you. I hope what you have done is of great help. So after this, now we are done with the quinolones. I've asked you to read and understand the mechanism of action of the quinolones. So after the quinolones, I think we are done. I think we will end the antimicrobacterial agents there in terms of TB. And now we look at leprosy. So leprosy, very few drugs, dapsone, and another one drug, very, very few drugs. I think lamprey, only about 2 or 3 drugs. So you can look at leprosy, draw the structures of the drugs that are used in leprosy, and then after that we go to the sulfonamides. So I don't think we are badly off. Then, of course, one day lincosamide is just a 1 day's job. So now next lesson, we should move on to Mycobacteria leprae. And as I have said, because again, it's a disease of they used to call us undeveloped world. Then you find that we have very few drugs that are there for leprosy, but nevertheless we look at them. Then after leprosy, after drugs for leprosy, then we look at sulfonamides, and then lincosamide, and we close the unit there. So I hope all of you have signed in. If there is any issue, please let me know. If there are any mistakes, please help me correct. Thank you very much. That if there are any mistakes in the structure, also, as I said last time, if you find a mistake in the structure, also look it up and also correct it at individual level, because these are structures, they are not a secret, they are there in the structures are also there in the website or in the internet. So you should be able, in case there is a mistake, you should be able to do the correction. So with that, see you. So we prepare for the cut on Friday.